Okay, that you can turn off. Okay, let me check.
लाइट 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 कर दीजिए ये वाला ठीक
Light glow. Okay. Okay, sounds good.
Okay. I think the Zoom audience is here. Yeah. I hope everybody can hear me. And it seems Professor G. Krishnamurthy is joining in the Zoom. So that's great. Okay. So I welcome one and all, both from Zoom, YouTube, and everybody here in this auditorium for a colloquium that is going to be given by one of our own, actually, Professor Samrat Mukhopadhyay, who was a part of TIFR almost 20 years back, almost 18 years back, actually. But before I introduce him, just wanted to say that it has been a fantastic week with Nobel Prize announcements. So today the chemistry Nobel was given to the quantum dots. So something that is absolutely relevant now in bioimaging as well. So going on to the NSF colloquium. So for those who are joining for the first time, the NSF colloquium, of course, as Samrat knows, this institute, it was started by our founding director, Professor Homi Baba, who used to come once probably in a week on a Wednesday to meet all the faculty members and discuss what's going on. And the faculty members would invite an eminent expert from around India or the world for a colloquium that everybody in the natural sciences would hear together. Something that I want again to be revived, right? Physicists, chemists, and biologists should be in the room today because the colloquium that Samrat has planned should connect to the physics as well as the biology audience, of course, the chemistry. Associated with the Department of Chemical Sciences in Aizer, Mohali. He has been there almost for 15 years now. He started his independent career right in that institute. But Samrat is kind of an interesting concoction of things in terms of the science that he brings. He was trained as an organic chemist from the Indian Institute of Science, Bangalore, with Professor Uday Moitra. And then he moved on to get introduced to proteins right here in this institute with Professor G. Krishnamurthy, who is hearing us right now. With GK, he spent 1 year, got introduced to proteins, and then moved to his position at Scripps Research Institute with Ashok Dennis, where he started looking at single molecule measurements on proteins and their very interesting folding transitions. Of course, Samrat's very varied background now puts him in a beautiful place where he's now pretty much the leader from the Indian community in terms of understanding the different forms of proteins, the conformations, and what other things other than function it does, as well as function it does when it makes protein condensates. So Samrat has, because of his wonderful work over the past 15 years, he is the editorial board member of the Biophysical Journal, which he holds very dearly, close to his heart. And he has won numerous awards. He's been the fellow of all the Indian Academy of Sciences and has been a very forceful proponent of looking into phase separations in protein aggregates, something that we call now as liquid-liquid phase separations, which has very, very significant and profound role as membraneless organelles in cells. So Without further ado, I would like Samrat to take over the floor and educate more about biological phase transitions, where chemistry and physics meet biology.
Thanks, Jyotishman, for a very kind and elaborate introduction. And it's always a great pleasure to be back at TIFR. And this place has played a very important role in my career. And as Jyotishman pointed out, that I was a visiting fellow about 18 years ago. And he has also stated that I was an organic chemist during my PhD from the Indian Institute of Science, Bangalore. And I was profoundly influenced by Professor P. Balaram during my PhD and decided to change my allegiance from organic chemistry to molecular biophysics. And at that time, I decided to have a short stint with Professor G. Krishnamurthy here at TIFR, which really changed my academic life and allowed me to really embark upon asking some very fascinating questions in the realm of molecular biophysics. And I would like to show you the evidence of the fact that I was indeed at TIFR 18 years ago. And this is a photograph clicked by GK's wife at GK's residence just before I was leaving for my postdoctoral training in the United States. I went to Scripps, and just before I was leaving, so it is in GK's house. And I have to mention that I was tremendously influenced and inspired by GK. And I was going, yesterday I went to the hallway of BG, that's the ground floor, labs BG30, where I really learned time-resolved fluorescence spectroscopy. And during those days, I collaborated with Professor Jayant Udgaonkar from NCBS, and that was really an important time. And it's Good.
अच्छा इसमें अनम्यूट करो.
Good. It's back.
No, okay.
Yeah. All right. Okay.
Yeah.
Some acharya is there. Yeah, that's, yeah, right. Okay.
Take it. Okay.
All right. So, okay. So I must thank really talented students I've had. My first student is now what happened to this? Okay. My first student is now a faculty at IIT Jodhpur, and several others are probably going to be faculty elsewhere. And the talk that I'm going to give today, the work was primarily carried out by Aishwarya, who is at Cambridge, and also Anamika, who has just headed to Scripps about a week ago, and by Ashish, Sandeep, and Anuja. So I'm also been thankful to my own institute and other funding agencies for generous support. So the next slide basically is a bird's eye view of the work that we have been carrying out for a little over 14 years at IISER Mohali. So we are interested in a class of proteins called intrinsically disordered proteins that challenge the tenets of central structure function dogma. So as textbooks have told you that a single polypeptide sequence can fold up into a single structure, and that three-dimensional structure is responsible for a particular function. And this particular dogma is defined by this class of molecules called intrinsically disordered proteins. They don't fold up autonomously into a well-defined structure. Therefore, they are capable of adopting a large variety of structures, offering a multitude of function from a single polypeptide chain, right? This is important for higher organisms, complex organisms. Chain. So that is that has an evolutionary advantage to expand the functional repertoire of proteins in the biological system. And of course, that comes at a price, because often this kind of structures can also undergo transition into states which are aggregation prone, which can undergo misfolding and cause a wide range of structures which are associated with diseases, primarily neurodegenerative diseases. And we have been trying to understand the conformational excursion of this intrinsically disordered proteins, which can happen on a wide range of time scales from grammar of chain-chain and chain-solvent interactions, which primarily drive the processes we are interested in, right? So we have used a multitude of technologies using ultra-fast fluorescence technologies and single molecule methods, vibrational Raman spectroscopic methods, coupled with a large number of imaging tools to try and understand the conformational excursion of these molecules from highly expanded to collapsed states, and ask the questions, what are the molecular determinants of these proteins to undergo transition into these exquisite nanoscale structures like oligomers, spores, and wide range of amyloid fibrils, and so forth. For the past few years, we have been immensely interested in another property of intrinsically disordered proteins, which is that they are capable of undergoing so-called liquid-liquid phase separation to form such condensed phases. Of course, these are all mesoscopic phases, okay, of the order of hundreds of nanometers to a couple of microns. And these condensates are responsible for the formation of what we call as membraneless organelles in the cells, and they orchestrate a wide range of functions within the cells. And you the formation of this beautiful multiphasic condensates, where the condensates phase separates within themselves to form multiphasic, multi-layered architecture, which is reminiscent of nucleolus, which is also a membraneless organelle in the cell. So let me tell you a little bit about this process. And of course, all of us are familiar with something called membrane bounded or membrane enclosed organelles like Golgi apparatus, mitochondria, and so forth. They're highly, localized and membrane enclosed. And in addition to these canonical organelles, the cells also possess non-canonical organelles, membraneless organelles, such as I said, nucleolus or nucleoli. Then you have got paraspeckles, female bodies, Cajal bodies, and in the cytoplasm, stress granules and P bodies and so forth. These are essentially formed via a process known as liquid-liquid phase separation. It is akin to oil phase separating from water. Or if you take vinegar, vinegar oil mixture, and if you shake it and then if you let it settle, then of course the droplets will phase separate from the bulk phase. So this is the process underlies the formation of some of these membraneless organelles, liquid-like organelles, because these are highly condensed phase. So basically, liquid-liquid phase separation is the demixing of a homogeneous phase of macromolecules in two coexisting phases. One phase is rich in macromolecules, and the other phase is, of course, depleted in macromolecules. So we can condense the macromolecules into a mesoscopic, liquid-like, highly dynamic, non-stoichiometric complex, such as these membraneless organelles. And you can recapitulate some of these organelles in an in vitro experiments. If you know the proteins and nucleic acids that are responsible for the formation of these organelles, you can use the purified proteins and RNAs or DNAs and to form such organelles in vitro and also in the cells, right? So, for example, you can see P bodies. Here you can see multiphasic architecture of the nucleus in the cell. This is the paraspeckles in the nucleus and so forth. Now, this is sort of like an interesting, analogy to lava lamp, right? So you have the droplets which are forming, dissipating. So this allows the biological system to have a control to form and dissipate depending on the demand of the cell. So these are on-demand organelles often, except I would say nucleolus, which is always present. But there are other organelles which are formed in a highly non-equilibrium fashion in response to cellular cues. And then, of course, they dissipate when cells don't need it. So these are on-demand, highly non-equilibrium process that goes on in the cell. So I'm going to say are this perfect liquid? Are this like, oil separating from water? Or there is some more complex behavior? It turns out that, hello? So it turns out that these are definitely not perfect liquid-liquid phase separation. As it is stated, these are essentially viscoelastic network fluid, okay? And the formation of such membraneless organelles are driven by density transition coupled with percolation transitions, okay? So these are system-spanning network compared to what one would assume as perfect liquid-liquid phase separation. On the next slide, I show you that many of these purified proteins or nucleic acids or together, you can take them and they can, create the situation which is there in the cell, and you can have this condensed phase, creation of this condensed phase in the test tube, right? You can actually make some of these things in the test tube, and you can study the formation, dissipation, and the other biophysical characteristics within these condensates, okay? Now, since it's a diverse audience, so I would also like to say, just introduce this, the essential thermodynamic driving force of phase separation, which is given by classic Flory-Huggins theory. Of course, all the details are not important. Here, of course, we have Helmholtz free energy, which is related to entropy of mixing and enthalpy of mixing, okay? And phi is essentially the volume fraction of the polymer. Notice there is this interesting parameter called Flory interaction parameter, which essentially, drives the enthalpic contribution of this relationship. So now, if you take, let's say, table sugar, okay, and you try to dissolve it in water, it dissolves, right? You take salt, it dissolves. It dissolves because of entropy of mixing, right? Because entropy of mixing essentially drives the dissolution, the mixing. But if you put these units in a polymeric architecture, the situation actually dramatically changes, because now in addition to entropy of mixing, you've got this enthalpic term, which is governed by the interaction, the chain interactions, okay? And that essentially counterbalance the entropy of mixing. So now you can, if you play around with the Flory interaction parameter and tune the enthalpic interaction, then you can have a situation where you can get past the entropy of mixing, and then you get a positive free energy of mixing. Under this condition, of course, it's positive free energy. Under that condition, mixing is unfavorable. Under this condition, of course, a polymer solution should phase separate, right? So this is the thermodynamic background of macromolecular phase separation, which has been known for several decades in polymer physical chemistry. And of course, you can get this kind of phase diagrams where you can place temperature and volume fraction in ordinate and absolute and get this beautiful phase diagrams, and that can tell you a lot about the phase behavior of the polymer. And of course, intrinsically disordered proteins are essentially a heteropolymer. But what really makes this protein intrinsically disordered is the presence of low sequence complexity. So you have 20 amino acids, but the sequence and the composition of amino acids are repetitive in this kind of proteins. Therefore, they don't fold up. Therefore, you have got this multivalent interactions exposed, because if the protein doesn't fold up into a three-dimensional globular form, all the non-covalent interactions are not satisfied. Therefore, all these multivalent interactions are now exposed. These multivalent interactions can now control the fluorine interaction parameter, and then allow the polymer, or in this case, intrinsically disordered polypeptide, to phase separate into a two-phase system, right? And what's the sequence grammar that you often see in this kind of proteins? You will see the sequences which are typically, described as a stickers and spacer architecture. So you have got certain amino acids which would like to stick to each other in an intermolecular fashion by using all the weak interactions like hydrogen bonding, hydrophobic interactions, electrostatic interactions, and so forth. And then you have got some of the amino acids like glycine, serine, and proline, which impart dynamism on the polypeptide chain. Therefore, this interaction, the sticker interaction lifetime, is actually ephemeral, and they don't last for too long because of the dynamism that is imparted by this highly dynamic amino acids. So if you have a stickers and spacer architecture, then what you get essentially is the formation and dissipation of the interactions. In other words, the making and breaking of the weak interactions, which give rise to the liquid-like behavior which we see. It's like water. Why water is liquid? Of course, the hydrogen bonds are stable only for tenths of picoseconds, right? So that the bonds are formed and then dissipated, and then it looks for another partner and forms another bond, right? And that's the reason it's liquid-like. And many of these biomolecular condenses, because of the making and breaking of interactions, you get the liquid-like behavior. Essentially, they are viscoelastic fluid, right? Now, I'm going to show you this particular result first. So liquid-like droplets or the condensates are highly functional. They carry out cellular biochemistry by concentrating the constituents of the cell, and it accelerates the cellular biochemistry. But often, some of these proteins, due to post-translational modifications or mutations, can promote these phase transitions from the liquid-like phase to gel-like or solid-like phase. Then they become like solid-like aggregates. They're highly pathological. So essentially, phase transitions are at the crossroads of physiology and disease. If you have more phase transitions, then of course it can go into pathological inclusions. And of course, if you have just the adequate phase separation, which is important for the biochemical reactions, then of course that's functional. Often the proteins that we work on, they stay as a liquid-like condensates in the nucleus where you've got this RNA. So nucleic acids actually, control the phase behavior of the protein by electrostatic interactions, and they form liquid-like condensates. And sometimes these proteins actually come out and form the stress granules. And if you remove the stress, these proteins actually go back to the nucleus. So that's the normal cycle. And many of these proteins we work on, they form stress granules in the cytoplasm. And then if you stress, for example, oxidative stress is withdrawn, then of course it goes back through the nuclear pore, which is also a membraneless organelle. It's a gel-like condensate in the membrane, which allows the selective passage of molecules. Now, if you have a mutation, then of course, these liquid-like condensates can undergo phase transitions into highly pathological aggregates. So I'll show you an example which we have been studying on here. Is a protein called fused-in sarcoma, a protein which is involved in neurodegenerative disease like ALS, amyotrophic lateral sclerosis, and frontotemporal dementia. Yes, here you go. So here you can see using a stable cell line expression, we are able to show this is del NLS represent for nuclear localization signal. Okay, if you delete the nuclear localization signal, it doesn't go back to the nucleus. It's a pathological mutation, and you can form this kind of beautiful condensates which are pathological actually because it can't go back to the nucleus. In the subsequent slides, I will show you how we study this under in vitro condition by recapitulating, by mixing things to form this kind of condensates we can study. Yes, Sudhita.
So you talked about the thermodynamics of it.
Yes.
That would describe the kinetics.
Right.
And if the cell has to use it,
Yeah.
the kinetics is extremely important.
Absolutely.
So how do you describe the kinetics? What is the characteristic of these things which will describe the formation and dissolution of these?
Right.
Are there theories on that?
Yeah. So
To talk about it.
right. So the kinetics of formation, of course, there are two kinds of transitions people talk about. Of course, one is binodal, okay, which is really rapid transition. There is also spinodal decomposition, which might take much longer. That requires a nucleation phase. And of course, many of these things can be sort of tuned by post-translational modifications, right? Post-translational modifications like the protein that we are working on, arginine methylation is a very important post-translational modification, including phosphorylation. So these are the two major PTMs in the protein that controls the kinetics of the phase transition in the cell in association with RNA, because RNA can also change the kinetics as well as the material property of the condensates, whether it'll be a liquid-like. Of course, liquid-like is a more sort of common man term or layman term, but I think we would like to see viscoelastic. Is it more viscous, less elastic, or less viscous, more elastic, which is solid-like? So it's sort of like a continuum transition from liquid-like to solid-like. So the material property and kinetics, these are controlled, highly governed by PTMs, so that's one of the, so in the subsequent, sure.
So do you think that has something to do with the physiological need
Yeah.
for the cell as
Yeah.
well?
Yeah.
Right?
Yes.
So-
Yes. So, for instance, if you need the membraneless organelles to function, for example, under stress condition, it has to recruit some of the proteins, very critical. It's a life and death for the cell, actually. Okay. It has to form the stress granule immediately. So arginine methylation, phosphorylation, okay, all this kind of, PTMs or post-translational modifications can actually speed up the formation of these membraneless organelles. And in this particular case, the stress granules, they recruit certain toxic substance which should not be exposed to the cytoplasm. And when the stress is withdrawn, then of course it can happily go back and form another class of membraneless organelles called nuclear speckles, and the function of which is not really known at this point. Okay, so in the subsequent slides, I'll tell you I'll give you some description about how we study some of these things under test tube condition in a very detailed manner using some of the very interesting tools, some of which we developed and we adapted some of these tools. Okay, first, I will talk about a protein which we have been quite interested in. Okay. For quite, oops. And this is a protein which you are incredibly familiar with, called the prion protein, right? It now appears in the 10+2 biology textbook. Okay, a protein which is infamously associated with mad cow disease and the human version of mad cow, like Creutzfeldt-Jakob disease, okay, deadly disease, a protein which is supposed to be in the alpha helical form, undergoes a drastic conformational change into beta-rich form, a protein called the prion protein. And then, of course, this beta-rich form aggregates into this kind of amyloid-like aggregates and forms these deposits. And this is sort of like the misfolded Mr. Hyde form. This is the Dr. Jekyll form. And the Dr. Jekyll goes to the Mr. Hyde. And then Mr. Hyde, which is the bad form, binds to the Dr. Jekyll form and stimulates the conformational conversion. And it is sort of like a self-perpetuating cycle. So if you have one misfolded molecule that can actually recruit, convert all the good form into the bad form, right? This is what prion protein is famous for, right? Now, often, we scientists make an implicit assumption that, the alpha helical part of the protein undergoes a transition to beta form, which is what is written in school biology textbooks, right? So the alpha part goes to the beta part, and that is what is the most pernicious conformational conversion. And of course, if you pay attention to the NMR structure of this molecule, you'll notice that the entire protein is actually not folded like it is displayed over here, because in those days, the NMR, of course, could not really capture the intrinsically disordered part. The half of the protein is actually intrinsically disordered, okay? And you don't see the entire N-terminal part of this protein because it doesn't show up. So the part that shows up is already highly disordered, and I have overlaid the NMR structures to tell you that the N-terminal part of this protein is actually intrinsically disordered. Now the question is, what's the function of this protein? Nobody knows. Okay, what's the function? Of course, the function is not to get misfolded and yield the disease condition, right? That's not the function. That's the dysfunction. So the function of the protein, of course, if you go back and find out what's the function, the functional segment of the protein is actually the intrinsically disordered part, where you will see oligopeptide repeat regions comprising a large number of histidine residues. And many of you know that histidine is a potent chelating, I wouldn't say chelating, but complexing agent. It forms coordination complex with cupric ion among the other ions, and it allows the copper homeostasis in the brain, because your brain actually needs very little amount of copper. So has to be a protein which will take cupric ion to the brain, which is needed, and this is the putative function. And there is an emerging consensus, this is the function of the prion protein. Now, how does it happen? Of course, as I said, copper homeostasis and coordination chemistry via the N-terminal disordered part. If you look at, I'll show you the sequence, one-letter sequence of this part, and which will tell you that it binds to cupric ion and also a little bit of zinc 2+ ion. Now, a number of years ago, I asked myself a question: if the intrinsically disordered part is what is responsible for function, then why there is this highly elaborate, intricately folded C-terminal folded domain, right? And this fold you are not going to see anywhere in nature. It only appears in the prion protein, okay? And of course, if you are interested later, I can tell you many esoteric features about this C-terminal folded part of the protein, including this is the only protein I know of which has more than 30 residue in a helix, longest helix that I have ever come across in a protein. There are many esoteric features, okay? Now, why nature has made this protein and retained it for millions of years of evolution during the vertebrate evolution? If you look at fish, if you look at mice, you look at dogs, you look at cow, you look at human, all of us have this prion protein. And we now know the function is perhaps located here, but biology is making tremendous effort to make this highly elaborately folded C-terminal part of the protein. This is essentially the question I asked many years ago. The answer is very difficult, right? It's a very difficult question to answer. Yeah, I'll come to that. I'll come to that on the next slide. Yeah. So now the clue came from a very intriguing, naturally occurring mutation of this protein, which is deadly. It's a disease mutation. You know what is this mutation? This mutation is called as amber stop codon mutation. So biologists would readily recognize this mutation. So there is a change in the DNA sequence, TAG, of course, the RNA sequence, where an amino acid residue, for instance, tyrosine, gets converted into a stop signal. So protein translation stops there, right? So there is no protein translation beyond that point. And I was so intrigued to see that the amber stop codon, mutation, pathological mutation, is at the interface of the intrinsically disordered and folded part of the protein, yielding only the intrinsically disordered part of the protein. Okay. And now, of course, you have been taught that alpha helix gets converted into beta sheet, but in this particular class of disease, which is a prion-like disease, which doesn't have the alpha helix at all. So the intrinsically disordered part undergoes transition to beta state, which is infectious, which is transmissible, and forms the basis of the disease. Now, if you look at the amino acid sequence, of course, you can readily recognize that there are many histidine residues in the N-terminal part. H stands for histidine. Okay, there are also tryptophan residues, weakly hydrophobic residues, and so forth. And here is Y145 stop, which is the mutation created here. And this is the C-terminal part of the protein. Okay, so now if we make this Y145 stop, then of course you get only the intrinsically disordered part of the protein. And we ask the question, what happens? What's the property of this intrinsically disordered segment? So Ashwarya discovered, of course, circular dichroism tells you that this is intrinsically disordered, as opposed to the full-length protein, which has the characteristic hallmark of the alpha helical protein, right? Now, if you raise the salt concentration a little bit, and this protein starts to form condensates, which is evident by the imaging experiments. Okay, and of course, this part of the protein, 1% of the protein is labeled using a green dye called Alexa 488, and 99% of the protein is unlabeled, and it is under super resolution microscope. You can see beautiful spherical droplets, and of course, are these liquid-like? And of course, yes, because they fuse. So of course, as you know, there are quite a few properties of liquid-like. They fuse and form larger droplets, which is a property because they minimize the surface tension. And of course, they are spherical. And then the mobility inside the droplets, the mobility of the molecules are actually quite mobile. How do you know that? Using an experiment called fluorescence recovery after photobleaching, or FRAP. So essentially what you do in the confocal imaging setup, at the center of the droplet, you crank up the laser power and photo bleach, you photo damage, and then you lower the laser power to look at the fluorescence. And of course, if there is considerable translational diffusion, then of course from the fluorescent part, the molecules will diffuse into the dark photo bleached part. And of course, the photo bleached molecules will diffuse out of that region. So you can get a recovery of fluorescence intensity. And using the kinetics of recovery, you can even determine the diffusion coefficient. And if you do all of that, you will realize this is actually liquid-like. Okay. And of course, there is a better way of measuring diffusion coefficient using fluorescence correlation spectroscopy, which we have also done later. So now the question is, the intrinsically disordered part undergoes phase separation and at a little higher salt concentration, because if you look at the sequence carefully, you will see, of course, the presence of histidine residues and quite a few lysine residues. So you have got this chain repulsion. In the presence of higher salt, there is a considerable screening effect, debihical screening, that allows now the polypeptide chains to talk to each other by hydrogen bonding, weak hydrophobic interactions, and cation pi interactions, and so forth, because there are also, positively charged residues that can participate in cation pi interaction, which we showed that is important for this kind of phase separation. Now, the obvious question arises, what happens to the full-length protein? This is what happens in the truncated form of the protein. What happens for the full-length protein? We were able to show that the full-length protein doesn't phase separate that well. In fact, there is something called saturation concentration. So beyond the saturation concentration, the protein will actually form the condensates. It's like dew formation. Beyond the saturation point, you will see the dew formation. Okay. So the saturation concentration for the full-length protein is much lower, and you can barely see a little bit of droplets here. And if you use this is actually turbidity plot, and you can also show along with turbidity the confocal super resolution image. So at that point, we propose that the C-terminal part of the protein, which is highly folded protein, which is essentially a regulator of phase separation that inhibits phase separation, and the N-terminal part is a promoter of phase separation. If you remove the regulator of phase separation, then of course it phase separates very readily, and it also undergoes transition into aberrant states like gel-like states, solid-like state, pathological states. Therefore, you have got the C-terminal part of the protein to regulate the phase transition. Fluorescence is actually great. It allows you to see beautiful images and all that, but it doesn't give you much of structural details, okay? All the exquisite molecular details, right, using fluorescence. So we actually resorted to the technique which we have been using and adapting for studying some of these protein condensates, called vibrational Raman spectroscopy, right? So essentially in Raman spectroscopy, you can look at all the bond vibrations, right? This is vibrational Raman. There's a scattering, it's inelastic scattering, happens due to vibrations, okay? That is also possible for rotation, but we look at vibration. So advantage of vibrational Raman spectroscopy is we can actually focus the laser into a single droplet, as you can see here. Okay, this is the crosshair, and this is where we focus the laser, where you've got the condensed phase of the protein, which is a droplet, and we can acquire the vibrational Raman signal, and then we can take the scattered light through the diffraction grating, and you can now ask the question, you can see every vibration, essentially, CN vibrations, CO vibrations, all the vibrations that are there in the protein, both the backbone of the protein as well as the side chains. And you can ask the question, what has changed? You can also go to the dilute phase and ask the question, what's the vibrational signature in the dilute phase and in the condensed phase? By doing many of these experiments, I am not going into the details of the results, we were able to show that under condensed phase, by using amide-1 and other vibrational markers, we were able to show that in the condensed phase, the protein is expanded. Protein, of course, this makes sense, because if the protein is expanded, then it is predisposed to interacting with other polypeptide chains in the form of multivalent interaction, which essentially drives the phase separation. Now, upon aging, these liquid-like condensates undergo transition, rapid transition in the time scale of several hours to days. Yes.
Sorry, Samrat, in the previous figure, I was not sure I saw what was the difference in the different, what peak was new in the condensed phase versus...
No, you will not see any appearance of a new peak. What you will see is, of course, the width of the distribution is different, and you can estimate from, these are single droplet Raman. So you can actually do Raman spectroscopy on a single droplet. And you can look at the amide-1, there is also amide-3 here. Amide-3 is more related to CN-NH. Amide-1 is more carbonyl. Then you have got this very interesting peak doublet here. This is tyrosine Fermi doublet. this tells you how tyrosine is exposed to solvent or in the protein. So you can use some of these markers to sort of construct the internal, architecture of the condensates. Okay.
Is
Now-
this saying that the vibrations are the same, or
Vibrations are same, but the ratios change. Some of these, of course, some of these intensity ratios and the width and the peak positions, they move around. And from the position of the vibrations, you can make certain structural conclusions about the polypeptide chain within the condensates or within the solid. Like I'll show you another set of spectra and on this slide. Okay, now upon aging, these liquid-like condensates undergo transition through this beautiful sea urchin-like morphologies to solid-like state. And of course, the frap recovery kinetics is gone because these are now gel-like, solid-like materials. So the molecules don't diffuse in the condensates. These are converted into solid-like aggregates. They don't diffuse now, which can also be looked at by Raman spectroscopy, atomic force microscopy. You can see fibrillar architecture. And you can see now there is a sharp amide 1 band which corresponds to amyloid-like fibrils. So now from disordered condensates, now it has converted into amyloid-like fibrils. And these fibrils have self-templating autocatalytic activity, which is a bona fide prion-like behavior, right? Because you can now take the seed and convert monomeric protein into more like amyloid state. Now we can, this part I can now conclude that basically my thesis is that, of course, you can have this protein misfolding from prion protein into this amyloid-like form. This is canonical misfolding pathway. Okay, some text is, whatever. Canonical misfolding pathway, which was proposed by Stanley Prusiner, which is what you have seen in textbook. And of course, I have got another alternate pathway, which is liquid-liquid phase separation, which is, of course, functional, which allows the copper homeostasis because cupric ion, coordination makes liquid droplets more pronounced. And these droplets perhaps allow the copper homeostasis, but then of course it has the risk of going into the amyloid-like state, which is prevented by this C-terminal folded part of the protein, which is essentially a regulator of phase behavior. So in nature, you have got now a promoter of phase separation and a regulator of phase separation. And that's how nature essentially controls, the functional phase separation. And in fact, this particular feature is now available in many of these proteins. We can observe this, including the protein that I'm going to talk to you about in the next 15-20 minutes. Okay. Okay, 10 minutes. Okay. Yes.
We can take a little bit more because
There was some disruptions, but I'll try to finish. Yeah.
So it's 350
Yeah.
million.
We have also used 150 physiological salt. Yeah.
And that concentration
Yeah,
also.
absolutely. Yeah. So you have to raise this. And of course, there is this, I'm not showing all the details. We can actually derive a two-dimensional phase diagram. You can have salt concentration, protein concentration. In the 3D, you can have temperature. So you can create a surface where the phase separation happens. So you can do a large number of experiments to sort of create this. Around a physiological salt concentration, little bit higher protein concentration at 37 physiological temperature, it phase separates beautifully. And very spontaneous. Within, like, you look at it, it phase separates. The kinetics, of course, I have not measured the kinetics, is faster than a second.
I have a question. So this full-length version, it does interact with membrane, right?
It's a good question. I didn't get into those complexity at this point. So there is a GPI anchor at the end of the C-terminal, which sits on the membrane. And the hypothesis is On the membrane, it forms on the membrane rafts. Of course, you can see membrane microdomain, cholesterol-rich region. It actually forms this kind of, puncta, which is probably a condensate. So you have a liquid-like condensate on the membrane outside the cell, which allows because that gets accentuated in the presence of cupric ion, and that cupric ion goes into the cell through that condensate. But now the problem is biology, if you want to phase separate, then you also need to have a strategy to lower the phase separation so that it doesn't go into the pathological state. And that's probably the strategy, evolved strategy by which the phase separation is controlled to the extent of just having functional implications.
Yeah, I was about to ask whether this truncated version also interacts with the membrane or not.
Truncated version would not interact with the membrane. Okay, there's a good question, by the way. I didn't, of course, discuss this. Truncated version doesn't interact with the membrane for two reasons. One, of course, the C-terminal end has the GPI signal. So GPI anchor happens at the C-terminal part of the protein. If you truncate before that, it is actually a cytosolic protein. It doesn't go onto the membrane. Okay, and it's actually a cytoplasmic, under cytoplasmic condition, it phase separates. Under normal condition, what happens? This protein, because the cellular machinery readily recognizes there is something wrong, so it chops off using proteasomal degradation machinery. But now if you inhibit the proteasomal degradation machinery, okay, and if you have a problem there, then of course it starts phase separating. And we have some results, unpublished results, to indicate that it might actually go into the nucleus. Okay, there is a very interesting hypothesis we have why it can go to the nucleus. So all this tug of war takes place within the cytoplasm. If you actually truncate it, there's one more truncation that can happen, which is also pathological, which we are studying. Okay, shall I move on now?
Yes.
Okay, so I'll have to be a little faster. So after this, we have also shown that prion protein can recruit other neuronal proteins like alpha-synuclein, like tau. They can also form what you call as electrostatic coacervates. So these are like electrostatically driven phase separation, okay, primarily electrostatic. And this forms like heterotypic condensate. So A interacts with B, it's like a binary mixture. Okay, A interacts with B, and that essentially forms an unit which basically phase separates. So it's essentially a heterotypic interaction, and these interactions can be modulated by introducing another molecule like ribonucleic acid. And this is something which we wanted to study because nucleolus essentially is a RNA and protein complex, right, which is involved in RNA biogenesis, and the multiphasic behavior of the nucleus is essential for cell functioning because you need to have the ribosome biogenesis. So in this particular, when we saw prion and tau forming this beautiful heterotypic condensates, we introduced RNA, which is now going to interact as the third component, which is going to interact both with PRP, prion protein, and tau, because both of these proteins have positive stretches. And of course, you have now got a matrix. A can interact with A, B can interact with B, C can interact with C, A can interact with B, A can interact with C, and B can interact with C. So you have got a huge interaction matrix between these components. And now, depending on the strength of these interactions, you can now phase separate within the phase separate condensates into prion rich or tau rich, depending on how it is interacting with the RNA. And this is exactly what we see in this particular case. When we add RNA, you see this beautiful transition from mixed condensates. Yellow means both green and red are present together. Now you keep titrating with RNA. We go to this beautiful core shell morphologies, to nested droplets, to inverse core shell morphologies. Okay, And then, of course, beyond a certain point, it dissipates, because if you introduce more RNA, there's charge inversion, and then it dissipates. So RNA essentially controls all the morphologies and material properties of these condensates that we were able to recapitulate in the in vitro experiments. In fact, in this particular slide, I'm going to do an experiment. This is there in the paper supplementary movie, which is one of my favorite slides. So I now have made with two proteins which are electrostatically interacting. I've made an electrostatic coacervate, okay, and I've added RNA, okay. Now, in the presence of RNA, as I said, it'll form this multiphasic condensate. So you have got more. One protein is rich outside, the other protein is rich inside, and they are phase separated. So you have got oil in water and two kinds of oil. So they are also phase separated. Now I wanted to do this experiment. This is in the presence of RNA. Now, if I add an enzyme like ribonuclease, which will chop off RNA, it's an RNA hydrolyzing enzyme. If you put RNA, ribonuclease, see what happens is I can now convert these multiphasic condensates into mixed condensates. Now they will become yellow condensates from multiphasic condensates, okay? And you can repeat these experiments over and over. So you could add RNA again, it'll phase separate and form multiphasic condensates. Again, you add ribonuclease, so you can do this repeating cycle and establish that you can recapitulate the ternary interactions. You can tune the ternary interactions to get to such multiphasic condensates, and you can study the kinetics of formation and dissipation of some of these aggregates, condensates. All right, so rest of the time I have, believe about 10 minutes I can take, or 5 minutes. So since it's a broad audience, I'm going to introduce two techniques, given the disruptions I have had, maybe 5 more minutes, a little bit more. Raman spectroscopy, I'm going to tell you two techniques quickly, which we have adapted, developed, whatever you can call. So Raman spectroscopy is a beautiful technique to look at, sort of decipher the, exquisite molecular feature. But the problem is Raman is highly insensitive. One Raman scattered photon for every billion Rayleigh scattered photon, because it's an inelastic scattering, very insensitive. And you can make it more sensitive, which will be very beneficial for such studies, because you can now, we want to do Raman imaging within the cells, within the nucleolus. How do we do that if Raman is insensitive? Because there are a couple of ways to make the Raman sensitive. Of course, Professor Sudipta Maiti has also worked on this particular technique. He has some really beautiful data on this. It's called surface enhanced Raman spectroscopy. So if you introduce nanoparticles, and on the surface of the nanoparticles, if you have a way to tether the protein non-covalently, then of course, if you use now laser, then there is a plasmonic enhancement in the near field of the nanoparticles, and that allows the enhancement of theoretically 10 raised to the power 12 enhancement, but for practical purposes, you can get enhancement from 10 raised to the 4 to 10 raised to the power 6 times enhancement in Raman signal. So this is a protein which I showed in the beginning, the images of the cell, called a FUS protein, which is an archetypal protein for phase separation. And you can see this orange dots essentially are the engineered silver nanoparticle clusters within the condensate of FUS, which is green fluorescent label. And we were able to show that this nanoparticles, which is engineered, can get readily, encapsulated into the condensates. And now if you do Raman spectroscopy, you can actually get huge enhancement. Of course, I'm not going to show you, and nanoparticles are at the center of the condensate, which I'm not showing using a Z-stack video. And you can show that are very low power, where you don't see any normal Raman, you can see beautiful surface enhanced Raman scattering and about 10 to the power 4 to 10 to the power 5 enhancement. So now I've been able to now turn an insensitive technique into a ultra sensitive technique, which will allow you to detect even single molecules using Raman spectroscopy, which is very important for looking at imaging in the cell by doing imaging of these condensates. And you can also study addition of RNA. You can do a mixed RNA, you can do Raman difference spectroscopy, you can look at the difference with and without RNA and ask the question, which part of the Raman spectrum have actually got changed, and that will allow you to answer the question that which part is interacting and altering the phase behavior. And the next tool I'm going to tell you is basically to look at using fluorescence. Of course, I said fluorescence is great, but it doesn't have the ability to give you the structural details, which is not entirely true. You can do something called Foster resonance energy transfer, which will tell you how far one part of the protein and the other part of the protein is. If there is a conformational transition, therefore you will see a different efficiency of Foster resonance energy transfer, abbreviated as FRET, right? Now, this is the same protein. We are looking at a low complexity domain of the protein. We can do selective labeling chemistry by using different kinds of chemistry. And now we have a way to sort of, we can also at some point incorporate our natural amino acid to look at this, to do selective labeling. And you can do this, steady state FRET. And of course, steady state spectra will not give you the information of individual molecules. Now, if you can do single molecule fluorescence resonance energy transfer, then of course you can study each of these molecule FRET of each of the molecules which is diffusing through a highly condensed laser volume, which is essentially the confocal volume, and you can capture what you call as fluorescence bursts. So when a molecule having the donor and acceptor pair is diffusing in and out through the confocal volume, then of course is leaving the footprint of its conformation by emitting the donor bursts and acceptor bursts. All you need to do for that particular burst, you estimate the donor acceptor photon numbers and get the FRET efficiency. So if all the molecules have the same conformation, then you will get a sharp peak FRET efficiency peak when you plot it against the number of events. But if they are highly distributed, then you can actually capture the conformational heterogeneity or the different conformational states, because if you look at millions of events, one molecule at a time, all this burst, donor and acceptor burst will allow you to separate the conformational subpopulations. And this is what we do using single molecule FRET. And we now have 4-channel detection. We can do 4 color measurements. We can do highly sophisticated single molecule FRET measurements, which allows us to look at conformational changes in the single droplet. So we can focus the lasers, actually 2 lasers in this particular case, into one single droplet and ask the question how conformation is changing. And if the summary of this work essentially tells you that in the monomeric dispersed phase, for this particular protein, you have an equilibrium distribution of a partially expanded tadpole-like structure and a compact paperclip-like structure in the monomeric form. And when it undergoes phase separation, you see there is a dramatic increase in the low FRET population, which tells you the polypeptide chain actually unwinds to participate in the multivalent interaction. So that's a key driver to phase separation, the unwinding of the collapsed conformation through, and then it can interact. Essentially, we're turning the intramolecular interactions into intermolecular interactions, which promote phase transition of this class of proteins. Essentially hydrogen bonding and pi-pi stacking are the major molecular drivers of phase transitions. So I will stop here, but on the last slide, I want to show something, particularly for the students. How many of you have heard of Alexander Oparin? Only a handful, right? Now, if I tell you, do you know Haldane-Oparin hypothesis? Everybody will raise their hands because you must have read Haldane-Oparin primordial soup hypothesis. In fact, this hypothesis got a little bit lost in the history. This is actually 1920s hypothesis. This because then Miller-Urey experiments of 1953, which happened around the time the DNA double helix structure was discovered, elucidated rather. The Miller-Urey did this experiment, and then that became origin of life, the best experiment. But now you go back and take a look at Oparin's hypothesis, who is essentially a Russian biochemist or Soviet biochemist, because in those days you had Soviet Russia. Okay, and he had published a book, The Origin of Life on the Earth. Okay, that is a 1938 book, and Oparin was almost almost a forgotten hero. Okay, and what he proposed was the life originated from organic coacervates. He did not call it as condensates; he called it as coacervates, colloidal coacervates, which first allowed the first biochemical reaction, prebiotic chemistry, to happen on the planet before even life was formed. Okay. Now, Oparin's hypothesis, of course, did not stand the test of time, simply because it did not account the presence of membrane enclosed cells. And now, with the emergence of phase separation, now it stands to, reason to go back and think about what Oparin did. And I want to say that it is very likely a life on Earth actually formed via phase separation. And that's why we are all here. Thank you very much.
Thank you, Samrat, with that profound statement. Questions for Samrat? Yeah, there are many.
And there is one question that she raised actually much before anyone. So you can decide. You are the chair.
Who was the? Oh, no, I'll come back to you.
So in your this surface Raman spectra, your particles that you are adding is changing the spectrum. So isn't that a problem? I mean,
so it is enhancing the spectrum.
No, it is changing the spectrum. If you can go back to the slide, I mean, the peaks appear at different places with different.
So there's only one spectra, one peak, which is essentially amide 1. It plays a little bit with amide 1. This is essentially because there is a selection rule, right? How that particular bond is oriented with respect to the surface of the nanoparticle. Therefore, you can have more enhancement and less enhancement based on the orientation of the molecule. But by and large, the peak position doesn't change much. Okay.
Yeah, I don't. So if I understood it correctly, it seemed that the whole spectrum has sort of.
This is usually okay.
Yeah, this plot is what I was.
Yeah, because this is a very weak. I've used. There's no spectrum because you have to.
If I look at that, let's say the red one and the green one, you see. I mean.
Yeah. So basically, what happens is, so this is amide 1. Amide 1 essentially becomes a little weaker here compared to the enhancement. And some of the peaks are not visible because there is normal Raman at a very low, in the concentration where we don't see, let's say, this power laser. Actually, we should be comparing these two. I showed this, that where you don't see the peaks here by using surface enhanced Raman, you see the peaks. But under higher concentration, if you do normal Raman, you can see most of the peak positions are actually similar. And some of the peaks you don't see. That's
actually, if you are saying they're similar, actually, I don't see a physical reason why they should be similar, right? I mean, it's on a surface, the molecule is interacting with the surface,
Yes.
The peak should shift. So actually, I'm confused, very confused.
So some of the peaks become less probable because, as I said, it's related to the orientation. Therefore, what happens is essentially the intensity of the peaks change a lot. Okay. but the peak position by and large remain the same unless they're very close to the nanoparticle surface, because it is not sticking to the surface. So you have about, I think, about 10 nanometer or 8 nanometer, in that range, you can actually get the, so any molecule which is oriented, let's say about 5 to 8 nanometer far, it can still get enhanced because it's still in the near field. It's not interacting with the surface. The cases where you have the molecules really stuck on the surface, you can see some changes.
My question was similar to his. How are you tethering these molecules to the
Yeah.
nanoparticles
So,
and the composition
good
and
question,
structure of
yeah.
these nanoparticles?
So we have spent quite a bit of effort in trying to tether this. This tethering is not covalent tethering. This is essentially an electrostatic tethering because you have got arginine residues on the protein, and on the nanoparticles, essentially we do negatively charged nanoparticles that attracts the protein. So when the molecule is, when the nanoparticle is inside, it is sort of trying to sort of take the proteins along, and then you have the illumination around the nanoparticle. So it's a non-covalent tethering. Pardon me?
What are these nanoparticles made of?
Silver nanoparticles. Sorry, I didn't mention. So we have done with gold nanoparticles and silver nanoparticles. And silver actually is much superior in this case in the enhancement. Although sometimes there is this oxidation is a problem. So gold sometimes works better for longer. But if you make freshly, silver works really well. No, these are nanoparticles, star-like nanoparticles. These are typically 20 to 30 nanometer. And what is the size of the droplet? The droplet size is of the order of microns. So they are very small compared to the droplets. They are forming around the nanoparticles? Yes. So basically, you can think in that way, or the nanoparticles go inside the droplets. Because of the negative charge on the nanoparticles, okay, it can get readily encapsulated in the droplets. And that enhances the proteins around the nanoparticles, and which is what you see enhancement. Okay.
Hi, Samrat. Beautiful work.
Oh, thanks.
Excellent work coming out of your lab. I have a question on the first part of your talk,
Sure.
phase separation. So now it is shown by your lab and of course many other labs all over the world that in general, intrinsically disordered proteins or proteins with intrinsically disordered regions generally can form phase separated droplets in biological systems under certain conditions. But there are a lot of molecules which are completely structured, especially
You
for
showed
the
one of those
function, right?
just
So
2 days ago, it was out.
Yeah. My general question is that, so what is the current understanding in the field
Okay.
in terms of functional molecules?
Yes.
How does this phase, separate phase, would have effect on their partitioning, stability, and function of folded or enzymatic molecules?
It's a very important question and very legitimate, especially at this point. So, so as I said, intrinsically disordered protein or a region of an intrinsically disordered region, which is basically sort of interspersed between folded domains, right? I mean, that is also a key driver. So you can have folded domains, but like in prion, you saw that is also true, but there are other proteins where you can have, sort of like a scaffold client relationship also where you need one more protein. But usually if you have a disordered state of the protein, you have the multivalencies exposed that allows, and of course, if you have a feature like sticker, spacer, that kind of feature, then you can get a liquid like condensates. Folded protein, the situation changes dramatically, okay? So one of the reasons a folded protein can phase separate, of course, in most cases, you will need much higher concentration to phase separate of 100 micromolar or of that order, whereas many of the proteins that we work on can phase separate in sub 10 micromolar, 1 micromolar, 2. Fast phase separates are 2 micromolar, for instance. There are other proteins where the saturation concentration is actually sub micromolar, okay? So that's one thing. And of course, that can be tuned again by post-translational modification. You can dramatically tune that. So folded proteins can phase separate if there are sticky surfaces. Okay, of course, one has to. So this is what, you can call this as patchy colloids, right? You have got the sticky surfaces, and of course, now you have got multivalencies. But in order to bring them together, you need higher concentration, sometimes 100 micromolar, sometimes hundreds of micromolar for some proteins. And you need to actually crowd the space so that they can't diffuse away. The only way to do that is to add PEG or dextrin or other crowding agents. Assuming that crowding agents will not interact with the protein, it will only sort of try to allow the proteins to interact. So that's one of ways of, getting phase separation of those patchy colloids. Okay.
Yeah, but I didn't really mean exactly the phase separation of folded proteins. For example, like you have shown nice example, RNAs. If you add RNAs, it goes into the liquid phase and then degrades RNA, right? So in terms of RNAs, does it specifically or preferentially prefers one phase over the other?
Yeah.
It is equally, it doesn't matter.
That's a very good question.
And
So basically what you are asking me to do, measure the partition coefficient.
Exactly.
Okay, which I haven't done.
One is partition coefficient,
Yes.
another is other properties like stability and function.
Function.
Of, for example,
In
RNAs in
fact,
your.
there are proteins now. In fact, one of my friends at IISc, Ashok Sekhar, works on a protein. He's also trying to look at structure and function within the condensates. Okay, and in the folded protein, another important thing is, which I think we discussed a little bit yesterday, you can also have transiently sort of unfolded state, because free energy of unfolding will tell you that 100% of the population is not folded. There are some populations which are disordered, right? Free energy will tell you that, equilibrium constant, and that you can crowd, okay? And that can introduce multivalency. And then you can recruit many of these things into so that equilibrium essentially shifts towards the condensed phase, right? So that's one of the ways to sort of think about the folded protein phase separation. Obviously, there is a structural transition, and you are taking advantage. And what you are asking another question, how it functions inside and there are enzymes now have been shown which functions very well in the phase separated condensates, because your diffusion is restricted and there is a huge reaction, acceleration of reaction rate.
Great, thank you.
Yeah, this is a quick question.
Yeah, sure. Good.
In your talk, you had a slide where you showed the time evolution. So what was the time evolution about? And did you measure the time evolution when this critical phenomena is happening? Is there any sort of divergence in the time scale? And the second question is, in the very beginning, you said that the transition is basically sort of percolation transition. So I was just thinking what kind of percolation is happening here?
So it's basically a networking, right? So what I was trying to say essentially, with Koti also, we had some discussion. I think you were also there. These are not perfect liquid-liquid phase. I mean, sort of like canonical liquid-liquid phase separation, right? Liquid-liquid phase separation is you take vinaigrette, right? Salad dressing, right? Shake it and then allow it to settle. You'll see the formation of droplets from oil, right? So that is liquid-liquid phase separation, because both are presumably, Newtonian fluid, right? In this case, the interior, as we described, in fact, some of our experiments, FCS, fluorescence anisotropy, many other experiments actually show that there are networks inside. So it forms sort of like a gel, you can call it as a gel-like network, although this is a system spanning network. Right? And that network coupled with density transition, which is probably a better description of biological phase separation rather than a pure liquid-liquid phase separation. Okay.
Okay. I think we will take more questions from students and other members in the coffee area, which we have arranged for all of you. We'll have some refreshments. So let's thank Samrat once again.
Thank you. Thank you so much.
I have questions that I will ask you.
Sure, sounds good.
Thank you, Samrat.
Okay, fine.
And I would like to thank the auditorium staff as well. I know it was a difficult day, but thank you.